This is Chapter 2, The Chemical Context of Life. So you may be wondering, this is biology class. Why do I have to study chemistry? Well, in the natural sciences, biology, chemistry, and physics are all kind of inherently linked together. So you can think of biology just as chemistry in action, and chemistry really is just physics in action. So if you go on to get a bachelor's degree in biology, you will have to take a few chemistry classes, so general chemistry, organic chemistry, and you should probably also have to take a couple of physics classes as well, because it's all going to be connected and linked together. So matter consists of chemical elements in pure form and in combinations called compounds, right? So all organisms are composed of matter. Everything is composed of matter, basically. So matter is anything that takes up space and has mass. Right? And all matter is made up of elements. Right? So we look at the periodic table of elements. This makes up all known matter in the universe that we know of. Right? So an element, these elements, is just one of the basic building blocks or substances that can't be broken down any further by normal chemical reactions. Right? So these are kind of our base building blocks for all matter on Earth and in the universe. So when we talk about a compound is just a substance that consists of two or more of those elements in a certain ratio. Compounds can have characteristics or some of those emergent properties that are different from its individual component elements, right? So we have sodium and chlorine gas. When we combine them together in a compound called sodium chloride, right now it has this different characteristic, this different structure, texture, right? These different emergent properties of table salt. Only about 20 to 25% of the 92 known natural elements are required for life, known as essential elements. So carbon, hydrogen, oxygen, nitrogen are going to make up 96% of all living matter. So the remaining 4% are things like calcium, phosphorus, potassium, sulfur, sodium, chlorine, right, salts. So these are the trace elements. So we only need these in very small, minute quantities. Each element consists of unique atoms that make up that element. So an atom is just the smallest unit of matter, but it still retains the properties of that element. Right? And then we can dive even further down and say the atoms are composed of subatomic particles, right? So sub being below. So below the atomic level, we have the subatomic structure. So this is kind of as far down as we're going to go. So the subatomic particles are neutrons. So neutrons are neutral, so they have no charge. Protons, right? So pro meaning positive, so they have a positive charge. And electrons. Electrons carry a negative charge. So protons and neutrons are going to cluster in kind of the center of the atom in the nucleus. So similar to like the nucleus of a cell, kind of in the middle of the cell. So protons and neutrons in the middle nucleus, and then electrons kind of orbit and hover around the outside of the atom. Right? Sometimes called an electron cloud. Right? So with these negative charges. So in terms of mass, neutrons and protons are considered to be one mass unit or one Dalton. Um, Electrons are so small comparatively that their mass is pretty much negligible. So it's very, very small. So usually they're ignored. They're not considered or calculated in when we're looking at the total mass of an atom. We just look at the protons and the neutrons. They're going to make up the bulk of the atomic mass. So atoms of various elements are going to differ in these numbers of subatomic particles. This is kind of how we determine where they fit on the periodic table. So an element's atomic number is going to represent the number of protons in the nucleus. So protons generally are also going to be equal to the number of electrons, right? Because protons having a positive charge, electrons having a negative charge, we want our atoms to be stable and equal. We don't really want them to be charged under normal conditions. So we'll have an equal amount of electrons to cancel out the positive charge from the protons. An element's mass number is the sum of the protons plus the neutrons in the nucleus. Okay? So if carbon has 6 protons, then it also has 6 neutrons, right? And remember, they each are going to weigh 1 mass unit or 1 Dalton. Right? So 6 plus 6, we get 12 for our mass number. The atom's total mass, or atomic mass, is almost the same as the mass number. So we're just including those electrons as well. So it'll have that slight little bit added at the end. But 12 kind of is our mass number. Atomic mass, the weighted average of the masses of all of the elements, subatomic particles. So isotopes are basically just different versions of the same atom. So all atoms of an element will have the same number of protons, but they could have a different number of neutrons. So isotopes are two atoms of an element that differ in the number of neutrons in their nucleus, right? So carbon-12 is our standard type of carbon. It's a stable carbon, so 6 protons, 6 neutrons. So its atomic mass is 12, right? So 6 plus 6 is 12. However, there is an isotope called carbon-13 where we have an extra neutron. Right? So now we have 7 neutrons. So now the atomic mass is 13. And we could also have a carbon-14, so it has 2 extra neutrons. Okay, so a lot of times these isotopes are radioactive or they're unstable, so they will decay spontaneously and can give off particles of radiation energy. Okay? So they want to become these stable forms of the atom, so they will eject these extra particles, these extra neutrons over a certain time period. So we can often use these radioactive isotopes for diagnostic tools in medicine, so radiography. Um, so radioactive tracers can be used to track atoms through metabolism. Right? So later on we talk about photosynthesis. The way that they kind of figured this out was using radioactive carbon and tracing that carbon atom through the pathway of photosynthesis. Um, we can also use it for sophisticated imaging instruments in the medical field. Right? So this is a picture of a PET scan that's monitoring the growth of different cancers in the body. Right? So it's able to light up those cells and those tissues, so we're able to detect them using this type of scan. We can also use these radioactive isotopes for radiometric dating to help determine how much time has passed since a fossil or a rock was formed. Right? So this is how we know, one way how we know how old fossils are, how old the Earth is. So as the original parent isotope decays into the more stable daughter isotope, this happens at a fixed rate. It's called the half-life of the isotope. So basically, different atoms have different half-lives. But say if it's 1 million years, right? So when this rock forms, it has only the radioactive form of the element. So one half-life time, so 1 million years passes. So at the half-life, half of those atoms have decayed. So now it's 50-50 of the original isotope and the new daughter isotope. So we can measure the ratio of these different isotopes and calculate, kind of go back, calculate how many half-lives have passed since the fossil or the rock was first formed. So another million years goes by, right? So now half of this remaining amount, right, is going to decay. So now we're left with 25%. Okay, so another million years, so another half-life. So half of 25% is going to decay, right? So now we're left with 12.5%. Right, another million years, right? So By measuring the age or measuring the amount of the radioactive isotope, how much is left, right, in terms of the daughter, we can kind of go back and see how many half-lives have passed, right? If we know how many years are in one half-life, then we can kind of determine how many years have passed since that rock or that fossil was formed. When we look at energy, we're talking about the capacity to cause a change or to put that matter into motion. So without energy, the matter, right, is not going to do anything. So we have to input energy into the system to make matter move, right, or do something. So there's two main types of energy. So potential energy is energy that all matter possesses just because of its location or its structure. So this could be considered a stored or an active energy. Okay, so in this example, the roller coaster, when it gets to the top of the hill, right before it moves for a split second, it's not really moving. That's when its potential energy is the highest. Kinetic energy is the energy of motion. So once that matter is actively moving, right, it has more kinetic energy. Right? So you can also think of the example of if I hold my pen up in the air, right? So right now I'm just holding it, right? It's not moving, but if I drop it, then it has, potential to move. So the higher up I hold the pen, the more potential energy it has, the farther it can potentially fall, the more it can potentially move. So different electrons of an atom can differ in their amounts of their potential or stored energy based on how far they are from the nucleus. Right? So again, the example of holding the pen up in the air. So the higher up I hold the pen, the farther it can fall, the more potential energy it can have. Right. So electrons, we said, were found kind of outside the nucleus, orbiting around. So they're found in what are called electron shells. Right. So each has kind of an average distance from the nucleus and energy level. So the farther away from the nucleus they are, the higher the energy of those electrons. So the chemical behavior of an atom is determined by the distribution of electrons in these electron shells. So basically, how an atom is going to react with other atoms. Right? So if it's going to form a chemical bond or reaction or not. So we use a periodic table to show our electron distribution for each type of element. Right? So we said kind of the ratio, the number and ratio of the subatomic particles are going to dictate where these elements fall on the periodic table. So when we read the table from left to right, each row is going to correspond to addition of new electrons and protons. Right? So lithium has one electron in the outer shell, right? Beryllium has two, boron has three, carbon has four, right? So on and so forth. So the electrons that are going to participate in our chemical reactions are called valence electrons. So these electrons are found in the outermost shell. So different atoms can have different numbers of shells, right? So here they only have two shells. Once the second shell gets full, we open up a third shell, right? And so on and so forth. So it's only the outermost shell or the valence shell that's going to participate in the chemical reaction. So think the inner shells are kind of insulated and buried under the outer shells. It's the outer electrons that are exposed that could potentially bump into other atoms and interact with other electrons. So this chemical behavior of atoms is going to be determined by the number of these valence electrons. Okay? So elements that have a full valence shell just naturally on their own, which would be 8. So helium only has 2 because it has 2 shells, but the other inert atoms will naturally have 8. So just remember 8 is the magic number. All atoms want to obtain 8 electrons in their valence shell. So they interact with other atoms to reach this goal of 8. When we look at chemical bonds, they're all trying to reach that magic number of 8. But some atoms already have 8, so they're considered inert or non-reactive. So they have 8 already. They don't need to react with other atoms. They're perfectly fine on their own. So sometimes these are referred to as the noble gases. However, most atoms don't naturally have a complete valence shell. So atoms with incomplete shells can transfer or share their valence electrons with certain other atoms to try to reach that goal of 8. So these interactions result in formation of chemical bonds. So this example, we have 2, 4, 6, 7. So each of these has 7 electrons, but they want 8. So they work together and they each share 1 electron. So now both obtain that magical number of 8. So one type of chemical bond is called a covalent bond. So this is where different atoms can share their valence electrons. In a covalent bond, the shared electrons count as part of each atom's valence shell. So carbon dioxide, CO2. So carbon naturally has 4 valence electrons. So it can share up to 4 other electrons. So the yellow ones are the carbon. So then it can share 1, 2, 3, 4 with the oxygen. So now the carbon has 1, 2, 3, 4, 5, 6, 7, 8 electrons. Carbon is happy. Oxygen, is shown in blue, only has 6 valence electrons. 1, 2, 3, 4, 5, 6. So it will share 2 electrons with carbon to reach 8. Right? So now all 3 of these atoms are bonded together by sharing their electrons. So they all have that number 8 in their valence shell. So we look at molecules. We're talking about 2 or more atoms that are held together by these covalent sharing bonds. Right? So depending on how many electrons are being shared, so we could have a single covalent bond. So they're just sharing 1 pair of electrons. We could also have a double covalent bond. So now they're sharing 2 pairs. So in O2, right, they're sharing 1, 2, 3, 4 electrons. So each oxygen is sharing 2 of its electrons together, right? So this would be a stronger type of bond. So we said earlier that carbon, hydrogen, oxygen, and nitrogen were 96% of all of the elements that make up living things. So we're carbon-based life forms. So if you've ever taken an organic chemistry class, it's all carbon-based chemistry. But what makes carbon so unique and ideal for these biological molecules is that it can form up to 4 different covalent bonds. So it can form a vast array of different shapes, molecular shapes, and molecules. So it can form 4 single bonds, it could do 2 double bonds, a double bond, 2 single bonds, a triple bond maybe, and a single bond. So it's very versatile atom. So we can have covalent bonds form between atoms of the same element or different elements. So we could have 2 oxygens bonded together to make an O2, right, or 2 nitrogens bonded together. So a molecule is just a general term for 2 or more atoms bonded together. So it could be the same or different atoms, any 2 or more is a molecule. However, when we specify something as a compound, this is a specific type of molecule that has 2 or more different kinds of atoms bonded together. So water, H2O, is a compound because we have 2 different atoms, hydrogen and oxygen, whereas oxygen, O2, is just a molecule. So one way to think of it is that all compounds are molecules, right? So they're all atoms bonded together, but not all molecules are compounds. So they have to have different types of atoms bonded together to be considered a compound. So I think a compound is more complex in its structure. So there are 2 types of covalent bonds we can see because the atoms in the molecule can attract or pull the electrons to varying degrees. So they're not, even though they're sharing the electrons, they're not always going to share those electrons equally, right? So it's kind of like a tug of war sometimes. So the electronegativity is just the atom's attraction for those electrons in that covalent bond. So how much pull do they have on those electrons? So the more electronegative the atom is, the more strongly it pulls the electrons towards its side of the molecule. So one type of covalent bond is a nonpolar covalent bond. So in these, the atoms are shared equally. So they spend equal amounts of time by both atoms in the molecule. Right? So they spend an equal amount of time near each nucleus. So nonpolar, think like the North Pole and the South Pole, right? So if it's nonpolar, it's equal, it's balanced. There's no one side pulling over the other. Okay, so they both have an equal kind of draw on those electrons. So these molecules typically are more linear or more symmetrical because, right, the electrons are shared equally. On the opposite end of the spectrum, we could have a polar covalent bond. So now we have one atom that is more electronegative, right? So we have an unequal sharing of the electrons. In this example, H2O, so the oxygen is a larger, more electronegative atom than the two hydrogens. So it's going to have a greater pull on those electrons. So in the tug of war, the electron tug of war, the oxygen is winning. Okay, so remember, electrons also have a negative charge. So whichever end of the molecule the electrons spend more of their time at is going to contain more of that negative charge. Right. So in polar molecules, right, so you have opposite poles. Instead of a north pole and a south pole, you'll have a slightly negative pole and a slightly positive pole. Ionic bonds are another type of chemical bond, but instead of sharing electrons, electrons are either going to be taken or gained, right, or given away. So atoms can strip electrons from their bonding partner. So atoms will lose or gain electrons. So instead of sharing, right, someone's going to lose one, someone's going to get another one. So the two resulting oppositely charged atoms are called ions. Right? So remember, electrons have a negative charge. So sodium is giving away an electron to chlorine, right? So sodium loses a little bit of its negative charge. So now sodium is considered to be the positively charged ion, or a cation ion. Okay? So it has a positive charge. Right? The atom that receives the electron, so in this case chlorine, so it's receiving an electron, it's receiving a little bit more of a negative charge, right? So it's going to be considered the anion. Right? And then opposites attract. So positive will be drawn to the negative charge, kind of like a magnetic attraction, which bonds these two atoms together in our ionic bond. Compounds formed by ionic bonds are typically salts or ionic compounds. So things like sodium chloride, just basic run-of-the-mill table salt. Right? So these are found in nature naturally as kind of crystal formations. So most of these salts are stable when they're dry, but they can easily dissociate and dissolve in water. Right? So think if you pour some table salt in water, it completely dissolves and you can no longer see the salt crystals or the salt particles. So what happens, we have our sodium chloride crystal. So the polar molecules of the water, so we have a positive and a negative. So the negative chloride ion is going to be surrounded by the positive end of the water molecule. The positively charged sodium ion is going to be surrounded by the negative end of the water molecule. So this is essentially what happens when something dissolves in water, right? So we'll talk more about water later and why it's vital for life. One of the properties of water that make it so vital for life is this polar nature of the water molecule, right? Its ability to dissolve substances like salts. Third type of chemical bond we'll look at are hydrogen bonds. So these are a bit different in that they're not true chemical bonds in the true sense of the word because there's no electrons involved. So hydrogen bonds really are just weak attractive forces between polar molecules. So we said water, H2O, is a polar molecule because the electrons spend more time near the oxygen than they do the hydrogen, right? So again, opposites attract. So the slightly positive end of one water molecule will be attracted through hydrogen bonds to the slightly negative end of another water molecule. Okay? So this is what makes water molecules want to tend to stick together, right, and provide surface tension. So if you ever kind of watch raindrops run down the window, and sometimes one raindrop will kind of merge into another raindrop, and they look like they're kind of attracted to each other, and they stick to each other and form a bigger raindrop. Okay, so that's those hydrogen bond attractive forces, are what allows insects, right, to sit on the surface of the water, are those hydrogen bonds, keeping those water molecules attached together. But again, these are weak bonds, relatively weak chemical bonds. It doesn't take a whole lot of force, right, to break through that water and to break those hydrogen bonds. Hydrogen bonds are also found in the DNA molecule. So DNA was a double-stranded molecule. Those strands are held together by these weak hydrogen bonds. So when we look at DNA replication, it involves the process of kind of unzipping these two strands. So we want a bond strong enough to keep the molecule intact, but we don't want to have to use a whole lot of energy to separate these two strands. So hydrogen bonds make it so we don't really have to use any energy. It's relatively easy to unzip and unwind the DNA molecule when we get ready to undergo DNA replication. Right, so summarizing the three types of chemical bonds, we had ionic bonds where electrons are transferred between atoms, so they either lose or gain an electron. These would be like your salts, like table salt. So the atom that loses an electron gains a positive charge called the cation. The atom that gains an electron is going to be known as the anion. So now it has a negative charge. And then these opposite charges will attract these two atoms to one another. Covalent bonds are sharing electrons between atoms. So a nonpolar covalent bond is where the electrons are shared equally. A polar covalent is where they're shared unequally. Right, so we have a slightly negative end where the electrons spend more time, a slightly positive end where the electrons aren't as frequent. And then hydrogen bonds are those weak attractive forces between those polar molecules, such as water. So touching back on the theme of structure reflects function, pretty much all levels of biology. So the molecular shape is going to determine how a biological molecule is able to recognize and respond to other molecules. Structure, the shape of something, can determine what it does and its function. So this example, our naturally produced endorphins molecularly have this shape, whereas man-made morphine drugs, opiates, have similar shapes, right? So it's very similar. So it's able to fit into our endorphin receptors on the brain. So kind of lock and key mechanism. So if you have the right shaped key, you can unlock, right, that lock. So because they're similar shapes, they're going to have similar effects by binding the same receptors in the brain. When we look at chemical reactions, we're talking about making and breaking of chemical bonds, right? So we're either forming new bonds, right, and sharing electrons or transferring electrons, or we're breaking bonds. So we're no longer sharing those electrons, or we're giving back the electrons. So the starting molecules or the ingredients of a chemical reaction are called the reactants. Right? So these are the ingredients, and the resulting molecules at the end of the reaction are called the products. Okay, so you have your ingredients, your reactants, and then you have your final end product. Right? So in this example, we have 2 H2 plus 1 O2. So we're going to break the bonds between these two oxygen atoms, break the bonds between these two hydrogen atoms, and then we're going to form a new bond between the oxygen and the two hydrogens. Same thing here, we have a CH4 and 2 oxygen molecules. So we split apart the 2 oxygens, right? We split apart the hydrogens from the carbon. So now we have a CO2, so carbon, right? We bond the carbon to the CO2, and then we have 2 water molecules. Right? So we've just broken some bonds and rearranged our atoms to form new molecules. The photosynthesis is one of the most important chemical reactions in biology. Plants are going to use the energy from sunlight, and they're going to use that energy to convert carbon dioxide from the atmosphere and water, right, from the soil, and turn that into sugar and oxygen. So in order to break these chemical bonds, it's going to require some energy, right, just to power this reaction, to break these bonds and form new bonds. Okay? So we get the CO2, and we cut off the oxygen. So for plants, oxygen is a waste product. Right, so they basically exhale oxygen. They don't really use oxygen, so it's a waste product for them. They just want the carbon from the CO2. So 6 CO2 and 6 H2Os, so 6 carbon dioxide, 6 waters, add a little sunlight, and we can rearrange these atoms to make glucose and oxygen. So glucose is going to then be used as an energy molecule to make ATP energy for the cell to carry out its life functions. It's also how we get nutrients from plants. When you eat plants or an animal eats some of those primary producers, they're getting some of that glucose energy that originated from the sunlight. And this is showing a plant undergoing photosynthesis and expiring or exhaling, if you will, little oxygen bubbles through pores in the leaf. Virtually all chemical reactions are reversible. So products of one reaction can become the reactants or ingredients for the backwards or reverse reaction. Right, so sometimes you may see kind of a double-headed arrow, just indicating that the particular reaction is reversible. Okay, so in this example, going forward, right, we have N2O4, right, so 2 nitrogens, 4 oxygens that we can separate into 2 separate NO4s. Okay? So we can also go backwards with that. Okay? Our previous example, we just talked about photosynthesis, right? So carbon dioxide, water makes glucose and oxygen. Well, animals, humans, we undergo cellular respiration to make energy. So it's essentially photosynthesis in reverse. So we take glucose and oxygen, that's why we have to breathe oxygen, and we break down sugar or glucose, and then we exhale CO2 gas. We make metabolic water, and then we use ATP energy. So where plants input light energy, we're going to harness the energy that originated from the sunlight, and now convert it into a chemical energy that our cells can use as ATP. So during chemical reactions, eventually, ideally, they will reach a chemical equilibrium. So when the forward and the reverse reactions occur at the same rate, so the relative concentrations of the reactants and the products do not change. So it's reached that equilibrium, that balance.